Hi everyone. Today we're going to bring you a topic which is known as the gastrointestinal system. So basically, this video is just going to be more about the overview of the gastrointestinal system. Okay, but I'll send a later video that is going to involve a detailed description or a detailed study of the gastrointestinal system. So basically, what comes to your mind when you hear the word gastrointestinal system? So these two things involve the study of the alimentary canal or the study of the GIT tract or the gastrointestinal tract, meaning it involves different organs coming together to perform a specific function, okay? Now it's being called a system. So we're going to involve the intestines, we're going to involve different structures such as the liver, we're going to involve the pancreas, you're going to involve the stomach. So it is going to house different types of organs that are working together in order to achieve a common goal or a common function. Okay. So now let's get to uh, what we call objectives. Okay. So now we should know that by the end of this session, you should be able at least to go away with something which has to do with the structure, the function, distribution, and mechanism of the gastrointestinal system. Why are we looking at this? So now, basically, when you look at this, you're going to understand that when you look at the structures of the JT, it's going to help you to understand where a certain mechanism is happening. Okay? Because basically, under physiology, you learn the normal function or the normal mechanisms that take place in a normal human being. So once, as a medical personnel, you understand how the normal body function works, it's easier for you to identify the anomalies or the abnormalities that come about, which are looked under pathology. So you need to start with the normal. Okay. So basically, we'll be looking at the normal structure, we'll be looking at the function, distribution, and mechanisms of a human being. Okay. Before we go to the pathology part or pathophysiology. Firstly, you're going to look at what we call describing the anatomy of the GIT tract. And later, we are going to discuss motility. So now, what do you understand by the term motility? So basically, motility has to do with how food particles are able to move from the mouth up to the anus. Okay, that's all part of what we call motility. Because motility simply means movement. So we'll be talking about the movements that happen in the mouth up to the anus. So that's what you're going to discuss under that. And if you look at the way they are arranged, you cannot skip that one and go to the other one because they are more like antagonistic. Huh? So, no, lack of a better word, they are simultaneous or they are linear. So you are going to find that if you look at one part, it will help you to understand the next session or the next subtopic. So if you look at motility, meaning, let's start with anatomy. If you look at anatomy, like I said earlier, it's going to help you understand how the structures are arranged. So once you understand where the stomach is, the small intestines are, what comes before the small intestines, what comes before the stomach, like the esophagus, etc., etc. Once you know that, then now you can apply the concepts of what we call motility. Okay, meaning you're going to talk about, okay, what type of movements happen in the mouth, all right? What type of movements happen in the esophagus? What type of movements happen in the stomach? What type of movements happen in the small intestines, etc. Just like that. Okay. So we're going to follow more like a linear pattern whereby we need to know one part before we go to the next. And then later, we are going to discuss what we call secretions. What do you understand by secretions? So basically, these are things which have to do with sorry about that. So now, when we talk about secretions, what are secretions? So these are chemical substances that are going to be produced in the GI tract or nearby organs, okay? Or we call them accessory organs. They are going to produce what we call secretions. And these secretions are going to enable or help digestion, okay? That's the purpose of these accessory organs, okay? They are not digestive organs, but they help to facilitate, okay, the GIT or the digestion and other processes that come about, right? So we are going to talk about different secretions that are going to be produced as we go along, and that will happen in the more detailed video that I'm going to make and send later, okay? So now, after you know the secretions that are produced, an example I can give you is salivary amylase. Another example I can give you is pepsinogen. Another example I can give you is saliva, okay, which contains different types of substances. Another thing that I can talk about is pancreatic juice, which also contains different types of enzymes like trypsinogen. Bile is also another example of secretions that we are going to discuss in detail later. Then after we understand the anatomy, we understand the motility, we understand the secretions, it's best now we can apply the concept of digestion and absorption. So as you know, what is digestion? So basically, digestion has to do with the breaking down of large, complex food particles into smaller ones with the help of chemicals called enzymes and other secretions. That is called digestion. Basically, you are reducing the particles from polymers to monomers. Okay? That's the basic concept of digestion. Then after that, these complex food particles have been reduced. They can now be taken up by the body. All right? That is known as absorption. Then later we are going to discuss about different types of disorders. In this video, I'll give an example of one of these disorders known as diarrhea. Okay? So diarrhea, basically, we've got two types of diarrhea. There's what we call secretory type of diarrhea and osmotic type of diarrhea. Okay? So under osmotic type of diarrhea, this is the diarrhea that is caused by you have an osmotically active particle in your intestines. So that osmotically active, an example of osmotic type of diarrhea is lactose intolerant. Those people are intolerant to lactose because they don't have enzymes that can degrade lactose, which is a sugar that is found in milk, and it's known as a disaccharide, okay? Meaning a dual sugar. So once you have that, you're going to find that that lactose, which is a disaccharide, meaning it has got two monomers bound together. So once you have that, you're going to realize that when the lactose is in the intestines, it's difficult for enzyme to break it down because these guys with that condition, they don't have this enzyme known as lactase. Hence, the sugar cannot be broken down, which causes water to move from the ECF into the intestines so that it can be able to dilute the sugar. Remember, it's just a concept of what we call osmosis or simple diffusion. In trying to dilute the sugar, hence you are going to have too much water in your intestines than normal, hence causing a watery diarrhea or stool. And that is defined as diarrhea because once you have passage of loose stool for more than 3 times in a day, it's going to be categorized as diarrhea. The other culprit, which is known as secretory type of diarrhea. An example I can give is cholera, okay? So cholera, when you have cholera, there are bacteria in your intestines, right? Which will be producing certain chemicals, which is going to cause an irritation. And because you have got a foreign microbe in your intestine, your body is going to have an immune response to try to wash away a pathogen, hence allowing a lot of water to move from the ICF and ECF into the intestines, hence causing you to lose a lot of water and causing you have allergic reaction like vomiting, etc., etc. Hence being described as diarrhea. So that's just a preamble or examples of what we call disorders of the GIT. So now let's go now to things that are more interesting. So like I told you today, we're just doing an overview. Okay. So now, overview. The gastrointestinal tract is a continuous tube that stretches from the mouth to the anus. Okay. So from the mouth up to the anus, that is categorized or that is described as the GIT. It includes the mouth, the pharynx, esophagus, stomach, small intestines, large intestines, anal canal, and anus. So all these parts that have been mentioned, they make up of what we call the GIT tract. It's a tube stretching from the mouth up to the anus. Okay, let's go to the third point. It also includes accessory organs, like I mentioned earlier. So these accessory organs basically facilitate digestion. Examples of these accessory organs are the salivary glands, the liver, gallbladder, and the pancreas. That are not part of the tract, but secrete substances into via what we call connecting ducts. Remember, when you go back, you're going to find that when you learn under endocrine system, you're going to find that we have got two types of, like for example, even the pancreas, we have got two types of systems that are there. We have got the endocrine system and we have got the exocrine system. Okay, so exocrine has to do with glands that have got ducts, exocrine, right? While endo, meaning inside, has to do with ductless glands. No wonder that belongs under the endocrine system, while exocrine falls under the digestive system. Because if you look at the secretions of all different types of enzymes and secretions, they are going to be secreted via duct, and then they're going to go directly into the area or the target that is going to be affected. While under endocrine, they're going to go into the circulatory system and be transported to areas where they're needed. Okay, but under the same GIT, as we go on, you're going to find that we are also going to look at what we call enteroendocrine, meaning the GIT also has got its own endocrine. Okay, it has got also its own nervous system, which is also known as the gut brain. Lack of a better word, gut brain. That's what it's called. Okay, or we call it the enteric nervous system, meaning you don't have conscious control what happens in the stomach, what happens in the intestines. It's beyond you because the brain has got its own, the gut has got, or the alimentary canal has got its own, which is known as gut brain, which can also be called by you educated people as the enteric nervous system. Okay. Anyway, there's also what we call carbohydrate digestion and absorption. So I'm going to go through 3 flowcharts, okay, to summarize this video, okay? And then later I'm going to send a more detailed video. So this is just a summary, like I said, or an overview, okay? So now, what you're seeing right now is a flowchart showing the stages that occur under carbohydrate digestion, okay? So basically, and absorption. Basically, in this video, we're going to look at 3 different types of macromolecules that are going to be broken down to smaller quantities, okay? So now, carbohydrate digestion and absorption. So there's action, foodstuff, enzymes, and site of action, foodstuff, enzyme, and site of action. So now, when you look at this, it's involving the stages. So now, what are we talking about? So we're going to start with carbohydrates. Amongst those 3 macromolecules, it's proteins, carbohydrates, and lipids. Okay? So these 3 things, that's the digestion we are going to look at. So now when you look at carbohydrates, give me an example of a carbohydrate. You can say starch, okay? So when you look at starch, is one example. So we can say starch and glycogen. But starch and glycogen, they're slightly different. Why? Because when you look at biochemistry, no one I always encourage my students that you need to know biochemistry, you need to know the anatomy in order for you to become a very, very good physiologist or a medical personnel. So when you look at starch, starch contains of what we call 1 to 4 and 1 to 6 glycosidic bonds. Okay, so if you are a student who knows biochemistry, you understand what I mean by 1 to 4 and 1 to 6 glycosidic bond. Okay, so starch has more of 1 to 4 glycosidic bonds, meaning the first and the fourth carbon, okay, bonding together in the glucose molecule. While glycogen has got more of 1 to 6 than 1 to 4. Hence, it forms a branch structure. So it's easier for your body to degrade starch as compared to glycogen. You're going to find that if you're breaking down starch, you obtain less of what we call oligosaccharides as compared to when you're breaking down glycogen. So you can go back and read, okay? You read also about amylose and amylopectin in biochemistry. You'll be able to understand better, all right? So now, after understanding, you've gotten a brief knowledge about the biochemistry of starch and glycogen, okay? So here's a situation, okay? You as a person or as a student, you take a lady out on a date. This lady buys pizza, maybe let me say something, something meaty or creamy chicken. So you're going to find that the dough has got a chicken there, or it has got meat there, there's cheese, there's what. So that pizza consists of the three basic macromolecules, okay? So when you get that pizza, starch will be part of the dough, okay? So now that starch needs to be broken down. So where does starch digestion begin? It begins in the mouth. When you ingest the food, the mouth will start moving, okay? The tongue will be involved, the teeth will be involved, and other parts that are involved. So that process is known as mastication, meaning the breaking down together with the action of the tongue to form what we call a bolus. And saliva plays a very important role. Why? Because it is able to lubricate the food so that it can flow, or it can move, or for easy passage down the esophagus. Okay? The voltage of esophagus, that's what we call the pharynx. So that's the purpose. But as starch is being mixed with saliva, there's a culprit, because in the mouth you've got about three major types of, salivary glands. There's the parotid, sublingual, and submandibular. Okay? So now, once you have that, you're going to find that starch is going to be acted upon by one of the enzymes known as salivary amylase or ptyalin. So once salivary amylase acts on starch, we are going to say digestion of both starch starts from the mouth. So once that happens, you're going to find that starch will be acted on by this enzyme known as salivary amylase, which is going to convert it to maltose. Okay, but as you know, if you go on a date, yeah, usually ladies don't consume a lot because they're afraid. But on the other side, the guy will be able to eat as much as he can. So you're going to find that the food won't take a longer period of time in the mouth. So the digestion will be temporal in the mouth. So as you swallow, it will go down along the esophagus. Okay? There's so many mechanisms that are happening there that I've just skipped. But when you look at a detailed video, detailed topic, you're going to realize that we are going to explain the stages of swallowing. So fast forward, the food goes in the esophagus. We have got the upper esophageal sphincter and lower esophageal sphincter. Their functions, we'll talk about them later. It goes straight into the stomach. In the stomach, so this is starch, and we have obtained dextrins, oligosaccharide, and maltose. Three things: dextrins, oligosaccharides, and maltose. For those who know their biochemistry very well, they know what it means when you say dextrins, you know what it means when you say oligosaccharides. So once you have oligosaccharides, meaning multiple chains, it has been broken down, but still has got many chains, about 10 or so carbon chains. That's what we mean by oligosaccharide. So now, once that happens, you're going to realize that when it moves down, okay, the esophagus, to the stomach, digestion won't stop there in the stomach. It will continue for like an hour before the enzyme salivary amylase gets denatured, okay, by the gastric juices, one of the secretions that is present in the all right. So I was cut off by network. Sorry about that. So now, like I was saying, it will take place in the stomach, okay? Digestion of carbohydrates just 1 hour before denaturing happens. So that's what happens. So during digestion of both starch, you're going to have these things: maltose, dextrins, and oligosaccharide. Okay, those are the things that you're going to obtain. So in the stomach, after the churning action of the stomach, then after gastric emptying, meaning the emptying of the stomach contents into the duodenum. So once in the duodenum, the stomach contents are empty there, there's going to be another enzyme which is going to be secreted by one of the exocrine glands in from the pancreas. Okay, that culprit is going to be called pancreatic amylase. That pancreatic amylase is going to continue where the salivary amylase left. Again, you are going to get the same things: dextrins, maltose, and oligosaccharides. Okay, because what was happening at the beginning was not extensive enough because of there was no time. It wasn't under conscious control. But now you are not controlling it. It's under the autonomic nervous system, or it's under the enteric nervous system. So it will take as much time as possible to release the contents known as maltose, dextrins, and oligosaccharide. So as the food continues down the duodenum, it goes into the jejunum, etc., etc. Then in the ileum, we are going to have what we call brush border enzymes. These brush border enzymes are able to produce what we call lactase, maltase, and sucrase, and alpha limit dextrinase. So these things are able to go and act on the lactose, maltase, and sucrase. Once they act on this, they're going to produce what you are going to see. Okay? So once they act on them, these enzymes, they're going to act. So lactose, like I was talking about in the beginning, if the enzyme that acts on lactose is known as lactase. So lactase is a sugar that's found in milk. So once it acts on that, we are going to produce what we call galactose and glucose. Maltose will be acted on, and then you're going to produce what we call glucose and another glucose molecule. Sucrose is going to be acted on by the brush border enzymes to produce what we call fructose and glucose. So this is the nature. Then the dextrinase are going to be acted on by alpha limit dextrinase, which is going to produce glucose molecules. So this is caused by a branched chain, 1 to 6 and 1 to 4. So 1 to 6 forms branch chains. So once it's acted on, it's going to produce also glucose molecules from the 1 to 6 glycosidic bonds. So once you have that, then they can now be absorbed into the secretory system. So for you guys who paid attention to cell physiology, okay, when you're doing your pre-med or you're doing your first years or introduction to physiology, you're going to understand that there will be a lot of mechanisms that has to do with cells. So if you don't understand, before we go in detail, please go back and study cell physiology or transport across the cell membrane. That will be very, very important on your part. So now, after this, let's now go and look at another culprit, which is going to be called protein digestion. So now, protein digestion, as you know, proteins are the most difficult to digest. Okay, they're the most difficult to digest as compared to carbohydrates and lipids. Lipids are the easiest to break down. So now, the same process, the same mechanism. So in the mouth, you're going to find that there will be the same anatomy that we described, the same motility that we described, but you're going to find that after mastication and mixing of whatever, there's nothing that happens in the mouth apart from physical digestion for proteins. Remember, the pizza, which we gave as an example, contains what we call. So protein digestion does not start in the mouth. It starts in the stomach. So fast forward, we skip to the stomach. Food has already reached the stomach, right? Or the bolus has already reached the stomach. So now in the stomach, you're going to find that action, protection, and absorption. Food stuff, the proteins. So now enzyme source, pepsin in the presence of hydrochloric acid. So I'll argue with this because once the food reaches the stomach, there's going to be an automatic response in terms of the secretion of what we call pepsinogen. Okay, so pepsinogen is going to be produced. There's going to be hydrochloric acid produced. So there will be, number one, the chief cells are going to produce what we call pepsinogen, which is the inactive form of pepsin. Then with the help of the secretion of hydrochloric acid, HCl, in aqueous solution, okay? So once it gets produced, it's going to cause the activation or the cleaving or the cutting off of the pepsinogen molecule so that you expose the active site, so that it becomes active. So once that happens, you're going to realize that the pepsin now, which was formed from pepsinogen, is now going to act on the proteins which are found in the stomach in an acidic environment, right? As you know, the normal pH for hydrochloric acid ranges from 2 to 4, okay, depending on the concentration. So now, once you have that, it's going to break down the pepsin, the proteins. So once pepsin acts on the proteins, it's going to convert these proteins into what we call peptones or just polypeptides. What's its major focus? Why does food go to the stomach? The reason why it goes, the digestion, food goes to the stomach and proteins need to be digested from the stomach is because it targets what we call collagen. So people who have got a problem with the secretion of pepsinogen have got collagen digestion problems. And as you know, collagen is one of the elastic proteins that we consume as human beings. Okay? That's one of it. So its target is to break down collagen, which is an elastic protein. So once collagen has been broken down, or it has been broken down to something much more softer, much more easier, then the food is, the bolus or the chyme, let's change the name. The chyme will continue to move because in the stomach, there's also gastric movements happening there, which is known as churning or gastric peristalsis. So after that, there will be gastric emptying. So the stomach, once they are emptied into the duodenum, the same mechanism that happened under proteins, under carbohydrates, you're going to find that the pancreas will release enzymes that are going to work on these proteins to produce different types of they're going to be acted by different types of enzymes, which are going to release different types of end products, okay? Or they're going to be reduced to simpler forms. Once that happens, that's what they're trying to talk about. So the pancreatic enzymes produce trypsin, chymotrypsin, and carboxypeptidase, also elastase, etc., etc. So all these are supposed to be in their inactive form. Then they're going to be later activated. That's what it's showing. So once that happens, so we are going to have small polypeptides. Then later we have got what we call the brush border enzymes. So on the side, it's showing where these things are happening. Don't forget, the brush border enzymes are going to have aminopeptides, dipeptides, tripeptides, all those things. They're going to act now on the things that came from the stomach, things that are coming from the duodenum, and reduce them further. So amongst all the different types of macromolecules that we are discussing, proteins are the most difficult to digest, and their digestion ends in the cells, inside the cells. We're going to see this when we discuss later. Then after that, they can now be absorbed into the secretory system and then transported to areas where they're needed. So now let's look at this, which is known as fat digestion and absorption. And you're going to end from here for this lecture video. So now, fat digestion and absorption. So basically, you're going to find that we do consume things that have got lipids. Remember the example that I gave you, something meaty or creamy chicken. I'm sure there's going to be some cheese somewhere, etc. Okay. So now that cheese contains what we call lipids. So basically, under this topic, we're going to discuss about 3 major types of lipids. We are going to talk about triglycerides. We are going to talk about phospholipids, and we are going to talk about what we call cholesterol esters. Okay. They also need to be broken down. So there's action, fat digestion. Why does this happen? So we have skipped. So remember, fat digestion happens in the mouth, it happens in the stomach, but now we are just have skipped and started from the duodenum. So as you know, in the mouth and in the stomach, it constitutes of about 10% of fat digestion. So now there's also what we call unemulsified fats. So now these are the unemulsified fats. We're talking about phospholipids, triglycerides, cholesterol esters. So all these, meaning that they have not been emulsified. So now let's look at the definition for emulsification. So what is emulsification? Emulsification agents that are being shown there on this table, on this, is bile acids, fatty acids, monoglycerides, lecithin, cholesterol, and protein. So now, these are things that go and emulsify fat. So what you're looking at right now is a fat is known as compound that's non-polar. Okay? Well, enzymes are polar, meaning they're able to interact with water. Okay? So fats are not water-loving, they are water-hating. Hydrophobic, while the enzymes are hydrophilic, meaning water-loving. So basically, you are trying to break down something that is hydrophobic using something that is hydrophilic. It's like a Jehovah's Witness marrying someone who's from a Pentecostal church. It can't work. Okay, it can't work. So one of the two, especially someone from the Pentecostal, needs to be converted, become a Jesuit or a Jehovah's Witness. That can work better. So now, emulsification, you are trying to, the fats are the Pentecostal. Why? While the enzymes are the Jesuit. So the enzymes want the fats to be converted into something that is non-polar. Even just pretending for the sake of people to be knowing that, no, my wife converted to becoming a Jehovah's Witness. But deep down, you know that they're not exactly Jehovah's Witness. So now we are going to come up with a lie, okay, which is known as emulsification. So basically, emulsification is the breaking down of large complex fats into smaller droplets with the help of bile and its ingredients or other substances, such as lecithin. So what is bile coming in to do? It's going to make the hydrophobic fats hydrophilic by breaking them down into smaller pieces. Once it does that, then it's going to form a fume of lecithin and bile salts and other things, so that on the outside, the fats can be able to interact with water in a better way, so that enzymes can now interact with the fats. Because as you know, water and fats are immiscible. They cannot be mixed. So once you do that, they are more likely to have been mixed because there's a line, a cover, a coat on the outside. forming tiny, tiny, tiny droplets. So that coat is now going to make the enzymes and the fatty droplets which have been emulsified to interact, hence causing digestion. So now, all the three different types of fats have got different types of enzymes. So now today I'm going to just focus on triglycerides. So triglycerides, as you know, they're going to be broken down by what we call pancreatic lipase. In the mouth, there's what we call salivary lipase. In the stomach, there's what we call gastric lipase. So now, in the duodenum, we are going to have what we call pancreatic lipase. Pancreatic lipase cannot digest the lipids without the help of emulsifiers, without also colipase. So once emulsification has happened, and there's also colipase, colipase is going to just go and anchor to the droplets and also anchor to the enzyme known as pancreatic lipase. Then begin the breaking down. Then we are going to produce what we call fatty acids and glycerol. That's what they're trying to talk about. Then they can now be moved by what we call micelles together with end products from phospholipids and cholesterol esters to the areas where they can be transported, or the brush border cells, where we have got also the lacteal. The lacteal is an extension of the lymphatic system, which is able to absorb the lipids and then transport them via the lymphatic system. And that's the end of the overview for the GIT. I've already talked about the pathophysiology. So later on, when we meet, we are going to talk about how the organs of the GIT are arranged, their importance and the anatomy bit by bit until we complete the GIT. Thank you very much.